Dear student, good morning. This is the second series, the second mini lecture in this module circulation. If you have followed on the previous lecture, actually it would be quite useful that you followed on the first mini lecture in this series, because there I described the basic concepts associated with the effects of gravity on the circulation. And this mini lecture is just the follow-up on the first one. But instead of concentrating on the brain circulation, on how come a giraffe can get blood up to the brain, in this lecture we are going to focus on at the other end of the animal. And this means basically the legs, the feet. And I title this on swollen legs because the main concept, the main idea that I fully explained in the previous mini lecture is that there are two problems that the giraffe has to face. Brain ischemia was dealt with in the first mini lecture. Now we are going to deal with fluid pooling or edema. And that is because, due to the effects of gravity, the expectation is that blood will accumulate in the legs. And because of this high pressure column, the expectation is that the pressure in the legs would be extremely high. And how can giraffes deal with this, which is the main concept of this lecture? Well, I have to revise some concepts of in relation with fluid leakage in the tissues. That's what we call edema, when this accumulation of fluids is enhanced, increases. And this is all based on a very basic concept, is that all capillaries leak. Some leak more than others, but all capillaries leak. And capillaries are the blood vessels reaching all the cells in all the small parts of the organism. So there are basically capillary networks everywhere. And all these capillary networks leak, some more than others. We have there are some capillary networks with continuous capillaries. They are the ones that leak the least. But then you have sinusoidal or fenestrated capillaries that leak much more. And this leakage is purely dependent on pressure. How that applies to our idea here is that the higher the central, or the higher the pressure, the larger the leakage. In the case of the giraffe legs, where the pressure is the highest that we can expect in any terrestrial animal because of the gravitational column, that is where we would expect the largest leakage, unless there are some changes, there are some adjustments that will prevent that from happening. As I said, even the tightest capillaries will leak, but then we have a system in the body that is responsible for dealing with this leakage, and that's the lymphatic system. The lymphatic system returns fluid to the cardiovascular system from this extra leakage in all tissues in the body. Of course, in the case of the giraffe, we have that the feet microcirculation has to see a transition of a blood pressure in the arteries at least 260 millimeters mercury to 150 millimeters mercury in the veins. So we have a very large pressure gradient that will be driving this fluid to move outside of the capillary network. And the question is how giraffes deal with that. If we only use hydrostatic pressures, the expectation would be that the giraffe would have highly swollen legs. And we know that that's not the case. Something is happening here, and that's what that I will be telling you in this mini lecture. First of all, let me revise the basic principles of transcapillary fluid balance so that you can understand basically what changes the giraffe has made and how that actually helps. But the first point is that the leakage is due to positive hydrostatic pressures in the capillaries. And that can be accounted, that can be quantified. If there is no positive hydrostatic pressure, there's not going to be any flow. Leakage, on the other hand, is opposed by another force. And that force is actually the presence of higher osmotic pressures in the capillaries because of proteins. So you have two essential forces opposing each other. One is pressure driving fluid outside. The other is the presence of proteins, the osmotic pressure of the blood holding on, keeping the fluid. And it's the fight, the battle, if you want, between these two pressures, the one that explains what eventually ends up outside of the cardiovascular system, outside of the capillaries. And this capillary, or this theory for capillary fluid balance, was formulated many, many years ago now by Ernst Starling. And his hypothesis state basically what I just said, that the net flow of fluid is based on the balance between the hydrostatic driving fluid out and the osmotic drive, or keeping fluid in. And this is what I will try to split up in the next couple of slides. Because the essential point, first of all, what forces are making loss of fluid? As I said, hydrostatic pressure, and this is because from the inside of the capillary, pressure forces water filtration, water loss. What's interesting, of course, is that this hydrostatic pressure changes along the capillary network. Changes along the capillary network because the blood pressure in this capillary network also changes as well. Closer to the arterioles, it is higher. Closer to the venules, it is lower. In the tissues, the hydrostatic pressure is considered constant. So essentially what we have is a gradient along the entire capillary. The gradient in the beginning, close to the arterioles, is larger, close to the venules it's smaller. In all cases it will drive the fluid, but it will be changing in quantity as we get closer to the venules. And this can be accounted for. If the plasma hydrostatic pressure is around, and this is given in international system units, 2.3 kilopascals instead of millimeters of mercury, and the interstitial hydrostatic pressure is around minus 0.73, that gives us essentially a pressure gradient of around 3 kilopascals. This is the force that will drive fluid outside of the capillary. If we look at the other part, which is basically which are the forces that allow for fluid gain or prevention of fluid loss, that means water reabsorption, this is the osmotic pressure. Essentially you have a constant gradient that is dependent on the osmotic pressures in both sides. Most of the osmotic pressure in the blood is due to proteins, and that actually amounts to a certain center pressure here. 73 grams per liter in plasma is the protein concentration in humans, and that basically gives a plasma colloid osmotic pressure of around minus 3.8 kilopascals. The interstitial colloid osmotic pressure is only minus 0.82. And the balance then in this case is a negative pressure, negative meaning that this is a reabsorption pressure, a reabsorption force that is keeping fluid inside the capillaries, of minus 2.98. Obviously, if we do the math, this generates a net pressure gradient of around 0.05 kilopascals. It's a positive gradient, meaning that actually this is fluid that is leaving out. If you do the balance along the capillary network, what you would observe is that you lose more, you leak more in the first part, close to the arterioles, and then you reabsorb more across the venules. But if you add both components, you still have a main leakage due to the hydrostatic pressure. And that's what it says here. In its passage through the capillary beds, there will generally be a net loss of water to the surrounding tissues. This is what transcapillary fluid balance theory stands for. What we need to do now is apply it to the situation in the giraffe. And in the giraffe, this is a very complicated slide. I'm not going to pay too much attention to it. I just want to say that to do these experiments, this was done by Hargens et al. In 1987. This required lots of measurements in lots of different places. The transcapillary fluid flow equation basically states a few things here having to do with lots of parameters in terms of the length, in terms of the pressures that are experimented here. The net fluid filtration pressure exactly basically can be calculated based on the hydrostatic pressures in both sides. When you do this math, essentially the calculations yield a net filtration pressure in the range of 88 to 152 millimeters of mercury. There is a considerable leakage, meaning essentially that at least from this study we don't see anything in the giraffe that seems to be particularly special about it. From the measurements of colloid osmotic pressures inside and outside and the hydrostatic pressure gradients, we would expect a relatively large loss of fluid. Does not look like the answer to the non-swollen feet in the giraffe is here. What all this indicates is that in giraffes we have a high susceptibility to edema. Edema that in humans is basically observed as this general swelling, that when you press on the tissue, there is this the formation of a concave. When pressure, basically it's a rebounding effect that takes a long time. So this is a tissue that is fluid filled. In all animals, and expectedly in giraffes as well, recordings of hydrostatic pressures during walking are highly variable. And that basically suggests that there is an effective mechanism, an effective pumping mechanism that moves blood against gravity. These and other aspects may make the difference for the giraffe. Which are these mechanisms predicting edema formation? The first one that we see is low capillary permeabilities to retain intravascular proteins. So what this is saying that essentially the capillaries in the feet of the giraffe are not of the most permeable type. We also see low tissue colloid osmotic pressures. The lymph colloid osmotic pressure is close to zero, and that essentially means that there are no proteins in the reabsorbed lymph fluid. But this is not the main difference that we see in the giraffe. Giraffes also have a prominent lymphatic system. They have one-way valves in veins and lymphatics, and all these will facilitate the return of fluid into the cardiovascular system and preventing it and leakage. And then we find some relevant changes that are quite different from what we see in most other animals. And this is this precapillary preconstrictions of the tibial artery. So when researchers have actually looked at the ultrastructure of the blood vessels in the legs of the giraffe, what they have noticed, this would be in the femoral artery, what they have noticed that at one point in the circulation in the arterial system down to the leg, there is an important constriction. It has even been called an sphincter. And that's what you see in this blood vessel here. You see the narrow lumen associated at this point, this would be the point number B, in the tibial artery. If you take a closer look at this, basically you would see this is a blood vessel with a very thick layer of muscle tissue that can basically constrict this. That's what has been called the sphincter. We do not know really why this is there, but the expectation is that this could work as a way to prevent excessive pressure in the leg circulation because of this constriction that's happening at this point. This is seen at this point high up in the leg, not later on. Notice. This is one of the particular anatomical difference that has been seen in the giraffe and has been not detected in any other animals. Later research has looked at the properties of these vessels, but relatively little of this is published still today. Finally, the other consideration is the fact that the skin in the legs of the giraffe is so tight, the tissues around the legs is so tight, that basically it would work like an anti-gravity suit, an anti-G-force suit works in fighter pilots. And remember, this is a very simple concept. In fighter pilots, the accumulation because of the accumulated G-forces they are exposed to, the accumulation of blood in the legs could lead to these people to faint. In order to prevent that, basically they are equipped with an anti-G-force suit that what it does, it compresses, in particular the legs, to prevent fluid accumulation there. So what we have in the giraffes is that essentially something that would look exactly like the same thing. Just simply that the design in the structure of the skin of the legs, everything is very compact and tight, preventing fluid accumulation in that sense. And that's the end of this lecture about giraffes, basically bringing these extraordinary animals, and in both mini lecture 1 and the second one now dealing with swollen legs, basically you see a set of adjustments at different levels in the circulatory system that allow these animals to defy gravity, that allow these animals to basically prevent the effects of gravity either by maintaining circulation, maintaining blood flow to the brain, or by preventing the accumulation of fluid in the legs. And that's the end of this lecture. Thank you very much for listening. I hope that you continue looking at the rest of the lectures in this module circulation.